Hi, my name is Roya and this is our enzyme experiment with Alex, Isabel, and Diksha. We are group 6D and my lab number. What? So basically what enzymes are they're just protein catalysts that increase the rate of reaction, and basically they speed up rates of reactions by lowering activation energy that's needed. And enzymes are really important because what they do is they basically, they just speed up essential reactions that are needed for human life that normally wouldn't really go as fast without them. And so basically, our enzyme that we've chosen to catalyze is trypsin. And trypsin is used to break down azocasein. And the role of trypsin in organisms is pretty much that it just digests dietary proteins. That's really what it does. And trypsin is active in conditions where it's found in the small intestine and it helps digest dietary proteins. It is found in basically neutral pH levels found in neutral pH, and our question and hypothesis is, how does temperature affect the function of an enzyme? And our hypothesis was, if the sample was placed in a warmer temperature condition, the enzyme would have a higher rate of reaction. And our rationale for the hypothesis was that while the enzyme has an optimum performance, enzymes have an optimum performance at higher temperatures because they increase the activity of an enzyme and the rate of reactions. So the conditions for our experimental variables were that the temperature variables were at 37, 50, and 60 degrees Celsius. And our conditions for pH was at a pH level of 9, and our salinity concentration is at 0.15 moles. And now for the steps of our experiment. The first thing that we did was to label all of the test tubes with the different temperatures that they were assigned. And all of the test tubes were filled with 450 milliliters of reaction buffer, 500 milliliters of azocasein, and 50 milliliters of trypsin. And each tube was placed in a different warm bath temperature with 37 degrees Celsius, 50 degrees Celsius, and 60 degrees Celsius. And the buffer allowed 10 minutes to incubate, and then we took out the tubes and added 500 milliliters of out of 500 milliliters of TCA out of each test tube to stop the reaction. And then next we centrifuged the test tubes at maximum speed for 3 minutes. So then after 3 minutes, we transferred the supernatant to clean cuvettes and up to a blank spectrophotometer at 440 nanometers to new cuvettes with water. And then we recorded the absorbances of our experimental groups once the spectrophotometer reached 440 nanometers. Okay, so then the reason why we added TCA to our experiment was in order to stop the reaction of the precipitation of the macromolecule. That's basically why. And then we had to centrifuge the TCA because we had to be able to separate the larger particles from the rest of the solution, and the pellet basically consisted of the enzyme and the substrate. And then azocasein was used for our substrate, and sulfanilamide correlates to the activity of trypsin. And then our control conditions were they basically included pH of 9, salinity concentration of 0.15 milliliters, and the pH of 9 was basically used because of research and the other observations from other group presentations. And then our negative controls were prepared pretty much the same way that our own experiment was prepared. And yeah, the same way that the experimental groups were prepared, but instead of using trypsin, we used water. And then we had 3 replicants because it was to reduce error in our results and to find any outliers that could have been in our data, and it was also just to measure variability. The average of 62 degrees Celsius was 2.9, and the mean variability, the average of 62 degrees Celsius was 2.209, and the variability was 0.011 for 50 degrees Celsius, and the average was 0.59, and variability is 0.088 for 62 degrees Celsius, and the average was 0.384, and the variability was 0.074. Our t-test results showed that our absorbance rate at 50 degrees Celsius and 37 degrees Celsius, as well as the absorbances at 50 degrees Celsius and 62 degrees Celsius are significantly different. And there was a significant difference in absorbance at a temperature between 50 degrees Celsius and 32 degrees Celsius with a value of 0.001, because if the p-value was more than 0.05, the significance would mean the difference is significant, so that we can conclude from our results that the null hypothesis is supported by 5% confidence, and our alternative hypothesis supported by 95% confidence.
Okay,
Our experimental design was adequate to test our hypothesis. However, our execution was not good. We had errors mainly with our water temperature in the bath and our spectrophotometer, and our hypothesis was partially supported in the experiment because it shows that temperature at 50 degrees Celsius is the temperature that trypsin had an optimum performance at. And compared to other groups, we were the only group that used temperature as our experimental variable, but we used a pH level of 9 and a salinity concentration of 0.15 moles because that's what other groups had used and said was the optimum for their experiments for trypsin. Yes, I think our choice of control variable conditions measured in the highest possible rate of enzyme activity, we maintained possible rate of enzyme activity. We maintained a pH of level 9 because 3 groups tested pH and the optimum level was 9 because trypsin had a better performance at a higher pH level. And a major source of variability that we had with the consistency was with the water baths. And this was a big problem with all of our trials because the temperature would often fluctuate, and so every trial it was different, and it really messed up our results. And going forward, to improve our experiments from first to second, we had increased a pH from 8 to 9, and we also increased temperature, and these changes allowed us to have more ideal conditions, and for future experiments it would be beneficial to make improvements to prevent error in our water baths. And ideal conditions of bacteria are normally warm temperatures because that, because that is what they thrive in. Warm temperatures increase warm temperatures increase the rate of growth. Increases the rate of growth and helps bacteria maintain homeostasis in order to survive and grow. And that's it. Thanks for watching.